Hello everyone, my name is Natria Mirelin. I will be taking you through the chemistry of lipids. So let's start. First of all, these are macronutrients that are needed by the human body. They play a very important role in nutrition as well as physiological functions. These lipids are rich in energy and are a very important source of essential fatty acids. They do not form polymers but are large molecules. They have low or no affinity for water, and for that case they are very hydrophobic. Now, these lipids have a long chain of non-polar covalent bonds. Most of all, or most importantly, is that the most important lipids in the body are fats, phospholipids, and the steroids. Now, let's look at the classification of these lipids. These lipids can be classified based on complexity, ability to react with alkali to form soap, and polarity. So based on polarity, or rather based on complexity, we can have simple lipids and complex lipids. The simple lipids include fats and oils. So the fats and oils are the ones we call triglycerides, and these are formed from triglycerols and fatty acids. We also have waxes under the simple lipids. These are fatty acids of long chain monohydroxy alcohols. So the second under complexity is complex lipids. These include phospholipids, and they also include glycolipids. So the phospholipids, these are the glycerophospholipids and the sphingophospholipid, as we shall be seeing. Then the glycolipids are basically, these contain fatty acids, the sphingosine backbone, and a carbohydrate. Others under the same category of complex lipids can be sulfolipids, aminolipids, and lipoproteins. Now, we can also have precursor and derived lipids. So the precursor and the derived lipids, these include fatty acids, alcohols, and aldehydes. So all those that we've been looking at fall under the category of lipids that are classified under complexity. Now we can look at lipids that are classified based on polarity. So based on polarity, we can have polar lipids and the non-polar lipids. The polar lipids being those that are soluble in polar solvents, like acetone, alcohol, and others. For example, we have phospholipids, glyceroglycolipids, and fatty acids. Now, the non-polar lipids, those are not soluble in polar solvents, so they are just soluble in non-polar solvents like ether, benzene, and mention all of them. For example, under this category, we have glycerides, sterols, sterol esters, carotenoids, waxes, and vitamins. So, we can also classify these lipids based on saponification. So based on saponification, we have saponifiable and unsaponifiable lipids. The saponifiable being those that produce soap after reacting with alkali. So they are always present in large quantities as glycerides, phospholipids, fatty acids, cholesterol, and esters. So the unsaponifiable, these ones do not form soap. So they include fat-soluble vitamins, sterols, carbonyls, and others. Now, all these classifications that we've looked at, polarity, based on polarity, based on complexity, based on saponification, they can all be categorized into two. That is, hydrolyzable or non-hydrolyzable. The hydrolyzable ones, these are waxes, triglycerides, and phospholipids. They are called hydrolyzable because upon, after being acted upon by the enzymes or during hydrolysis in presence of an enzyme or an acid, these are capable of forming triglycerols, and they are capable of forming their constituent compounds. Basically, for example, triglycerides can form triglycerols and the corresponding fatty acids. Same applies to waxes and phospholipids. All these are esters in nature, so upon hydrolysis, they form their constituent compounds. Now, we also have the non-hydrolyzable lipids. These ones include steroids, fat-soluble vitamins, and eicosanoids. These three, these ones cannot be acted upon, rather they cannot hydrolyze to give their constituent elements, a reason as to why they are called non-hydrolyzable. So these lipids, while present in the body, they play two important roles. One is the physiological function, and the second one is the dietary function. So to begin with, let's look at the physiological function. Now, when we look at these fatty acids, they are components of other classes of lipids, and they also act as metabolic fuel in the body. So the triglycerides, these are the main storage forms of fatty acids, and they also generate ATP or energy for the body. Now, the phospholipids, these are components of membranes, and they are responsible for, or they are important for, signal transduction. So we also have sphingolipids. These are also components of membranes, and they are also important for signal transduction. The cholesterol is a component of membrane, and it is a precursor for bile salts and steroid hormones. Okay, so we also have bile salts. These are very important in lipid digestion and absorption. We all know that lipids are hydrophobic majorly, but the body is always aqueous. So the bile salts help in emulsification of these lipids to enable the digestion of them to take place by just breaking them down into small micelles. Now, we have the steroid hormones. These are intracellular signals that regulate gene expression in the target cells. So, another important lipid is they are the eicosanoids. Specifically, we look at prostaglandins, thromboxanes, and leukotrienes. These regulate both physiological and immunological functions of the body. Now, lastly, the vitamins. These are A, D, E, and K. We know them as important for vision and calcium metabolism. They can be antioxidants and also help blood coagulation. So those are the physiological functions of lipids. So the dietary functions, these lipids are a source of energy. If you compare them with carbohydrates and sodium, for every gram of lipids, of fats and oils that you consume, it will give you 9 kilocalories, unlike the carbohydrates, which contribute only 4 kilocalories, and proteins also 4 kilocalories per gram. Now, these lipids also are carriers of fat-soluble vitamins. They are very good sources of essential fatty acid, such as linoleic acid, alpha-linoleic acid. They are also very good in a way that they impart taste and flavor to food. If you're eating food that is fried and food that is boiled, you can easily tell a difference. The other one is a little bit flavored and tastes good. And so they are very good lipids in terms of organoleptic properties. Now, raspberry is the satiety value. When you eat food that is rich in fats and oils, you feel full. You feel so full, so fast compared to someone who has eaten unboiled and fried food, or food that contains those fats or oils. Now, so those are the two basic functions of the lipids. Now, if we are to look at the simple fats from the classification, we are going to begin with the triglycerides. The triglycerides are formed from a reaction between glycerols and fatty acids. So you have a triglycerol reacting with a fatty acid to form a triglyceride. So this is like comparable to a simple esterification between an alcohol and a carboxylic acid, whereby each hydroxyl group on the triglycerol can react with a different or similar or a carboxylic or a fatty acid to form an ester while it is releasing a water molecule. So these can be replaced by three molecules as seen here to form triglycerides. So the triglyceride basically contains a glycerol backbone and side chains of fatty acids. These fatty acids can be similar. Now, this is an example. This is a triglycerol backbone, and these are three fatty acids. He said they can be similar, they can be different. Like here, they are just different. Now, most common lipids, both in food and in the body, are actually triglycerides. So they make up about 95% of lipids found in food. And that's why we are paying more attention to these triglycerides among other glycerides. Yes, we don't only have triglycerides, we have also diglycerides, and we can have a monoglyceride, the one that contains one glycerol, three glycerol or three alcohols, or is a triglyceride, the one that contains two. It is called a diglycerol. Okay, but the triglycerides are the most common. Now, how important are these triglycerides? First of all, they add texture to food. Two, they make meat tender. They can preserve freshness in food. They can also be stored in the adipose, part of the body. Just in case energy is released, they can be broken down to release some energy. Now, these triglycerides can be simple or mixed, whereby the simple ones contain the same fatty acid. The fatty acid that reacts with the OH group of the triglycerol is the same. In other words, you can have three fatty acids that are similar, or the chain is the same, the length of the chain is the same, and when they are different fatty acids are reacting with the triglycerol backbone, like you can see this one in red, this one in green, this one in blue. In terms of the chain, in terms of whether unsaturated or saturated, they can be different, and so they have to form a mixed triglyceride. Now let's look at the physical properties of triglycerides. First of all, triglycerides, when we talk about them, we are simply referring to a fat or a solid. Okay? So if a triglyceride is a solid at room temperature, it is called a fat. If it is an oil at room temperature, it is called an oil. Now, these triglycerides that we are talking about that are obtained from animal sources are usually solids, while those obtained from plants are usually oils. Okay, so that's why in most cases we talk about animal fats and vegetable oils. Now, crystal formation. Triglycerides can form crystals depending on their symmetry or the similarity of the fatty acid in the chain. So if, for instance, the fatty acids in the chain are the same, and let's say they are saturated, okay, you find that they align themselves properly, and so because of the compactness of the molecules, they can form solids. Okay. Now, polymorphism. The triglycerides have a tendency to exist in different forms, and this will depend also on conditions during crystallization. Okay, the composition of fat. The melting points also of these triglycerides will depend on the heterogeneous nature and the length or isomeric configuration of fats. So look at all these a little bit later in detail. Now, density. When you talk about the density of these triglycerides or of any substance, you're simply referring to how much of the material can fit in a particular volume. Now, this will depend in these triglycerides. The density will depend on the efficiency of the packing of the triglycerol molecules or triglycerides. If they are not so packed closely, then they will occupy a bigger volume. Okay? And so you can tell from their density. Now, these fatty acids that we are looking at can vary in length, they can vary in saturation, and can vary in shape. And because of that, we can have different triglycerides. Remember, the backbone of these triglycerides does not change. It is only the fatty acid that is changing. And for that case, we shall pay more emphasis to the fatty acids. So these fatty acids are also building blocks in phospholipids, as much as they're also building blocks in triglycerides. Now, most of these fatty acids that are used to make triglycerides or that form triglycerides are of even carbon number. You never find that you have maybe a fatty acid or maybe say 5 carbon atoms. No. So they can range from carbon 2 to carbon 26, the most common ones. So we are saying that in straight chain, even number carbon with a carboxyl group, and in most cases, not actually in most cases, most of these fatty acids, all of these fatty acids are actually monocarboxylic. They contain one carboxyl group. So this is an illustration. This is a methyl group. This is the chain of the carbons present, and this is the carboxyl group. So we said fatty acids can vary in length, and for that case we can have short chain fatty acids These contain 2 to 4 carbons, and in most cases there are weaker forces of attraction in between these molecules, and because of those weaker forces of attraction, they are liquids at room temperature. We also have medium chain fatty acids that have 6 to 10 carbons. They are also there. We also have longer chain, which have from carbon 12 to carbon 20, and they are the most common in foods. They have stronger forces of attraction. Why? If you go back to basic chemistry, the higher if the molecular mass is really high, then even the van der Waals forces of attraction, the strength of the van der Waals forces of attraction also increases. And for this case, it is justifiable to say that they have stronger force of attraction, and because of that, they are also solids at room temperature. Now, the very long chain of fatty acids can be from 22 carbon atoms and above, but we are saying that the most common ones are from 12 to 20 carbon atoms. We also said fats can vary in saturation, meaning we can have saturated and unsaturated. So all the carbons in the saturated fatty acids are actually bonded to 4 other atoms, and they are solids at room temperature. They are also high in melting points because of that kind of arrangement, proper arrangements, and their fats in most cases are solids at room temperature, like we said. We said because these fatty acids are saturated, therefore the fats are saturated fats. And the most common occurring saturated fatty acids are myristic acid, palmitic acid, and stearic acid. When you look at these figures in brackets, they shouldn't scare you. So the 14 here, just in case the carbon chain or the number of carbons, this 0 in brackets indicates that there is no double bond. So since the carboxylic group is ionized, it can, from just basic chemistry, this is a carboxylic acid, it can ionize, or it can partially ionize, release the hydrogen ion found in this an ion, the carboxylate and the hydrogen ion. That's what we have in there. These arrows are supposed to be up here. This is the fatty acid chain. After it has lost the hydrogen ions, it forms a palmitate. If it is from palmitic acid, myristic acid, you know what it can form. So this is just an illustration. These are 2 oxygen molecules. Now the unsaturated ones, these have double bonds in them, so we can have the morphers. So I have to edit this. This is supposed to be a morpher, monounsaturated fatty acid. Okay. We also have puffers, supposed to be a morpher. Okay. Then we also have a puffer. A puffer is a polyunsaturated fatty acid, and it can have 2 or more double bonds. So examples, we have a linoleic acid. We can have alpha linoleic acid. We can have arachidonic acid. So when you look at the brackets, Okay, if when you look at the brackets, these ones, not the brackets, these figures, you can see 2, 3, sorry about that. So we said you can have, sorry about that. Yes. So this is 2, indicating there are 2 double bonds, 3, there are 3 double bonds, 4, there are 4 double bonds. Now the unsaturated fatty acids, these are liquids at room temperature. Why? Because they cannot align so well. So there are so many spaces in between the unsaturated ones, which makes them to exist as liquids at room temperature, and for that case they will have low melting points. So fats made from unsaturated fatty acids are called unsaturated fats or oils and are majorly liquids at room temperature. So plants and fish oils are usually unsaturated. Now this is an example. This is a saturated fat and this is unsaturated fat. So when you look at a saturated fat, this is a solid and this is a liquid. Why is it a solid? Because it has saturated chains. There is no double bond. And if you look at these molecules, they will be in position to align very well compared to this unsaturated part, which will make them to be a little bit further spaced, and so they can exist as liquids. Now let's look at fatty acid compositions of food fats. Now most natural fats, such as those in vegetables, oils, dairy products, mention all of them, okay, are a complex mixture of simple and mixed triglycerides. Like, you may not say that they contain basically only simple, or they contain only mixed, rather saturated one. They can contain both saturated and the unsaturated. And the saturated can be long or even medium. Now because of that, they will have various or varying physical properties. And as we can see from this diagram, this is olive oil, this is butter, and this is beef fat. So this is the fatty acid content. So we are saying that the green one is the unsaturated, then the green one is the saturated, and the yellow one is the saturated, but having the medium carbon chain, which is from carbon 4 up to carbon 14. So if you look at this, olive oil is able to exist as a liquid as much as it has saturated and unsaturated, because the total concentration of the unsaturated part, or the percentage of unsaturated fat, is bigger, it will exist as a liquid. Now butter is a solid and beef fat are also solids, but the bigger the unsaturated part, okay, that we are seeing here in beef fat, is the one making it exist as a hard solid. Because as much as we have this as a saturated, we also have the highest, a bigger percentage of the saturated medium acid, and also the percentage of the unsaturated is also big. So because of this, it can be a solid, yes, but because of this, it will be a soft solid. But here, because the percentage of unsaturated is bigger and long, then it is going to be a hard solid. So this is an example. So the fat acids in the saturated fat are very, very aligned together. There's a small space, whereas here, because of the double bond, there is bigger spacing, so which makes them less packed, and so they exist as a liquid, whereas these ones that are very closely packed exist as solid. So we said fatty acids can be unsaturated, but even in their saturation, they can also differ in the double bond location. In this case, we can look at omega-6 and omega-3. For example, the location of omega-3 is at position 3. We count from the omega side, the methyl group. So the position where you see the first double bond is where you stop counting. So from here, 1, 2, 3. So this becomes omega-3. Then these are the foods rich in it, and an example is alpha-linoleic acid. So we can also have omega-6. If we count from the omega side, this is carbon number 6, so this will be omega-6 fatty acids. So we can have omega-6 and omega-3 fatty acids, and these are the foods rich in those fatty acids. Now, fatty acids can differ in shape and geometrical isomerism. For this case, we can have the cis and the trans. Okay? So when you look at the cis, the hydrogen atoms are located from one side, whereas in the trans, they are in the trans direction. And because of this, we shall find that the trans has a higher melting point compared to the cis because they can align so well. You can see the figures here. This is still the same stearic acid, but in the cis, it has a double bond. This is an unsaturated one, and this is also unsaturated, but here the double bond is trans, and here the double bond is cis, so you can see the values in the table. Now, these trans fatty acids are literally not so good, and there are studies trying to study more of these. Actual studies have found out that the trans fatty acids are more dangerous and can lead to increase in plasma control cholesterol levels compared to the cis isomers. And because of this, the current generations are trying to remove these trans fatty acids from the oil or from the fats. So the current concern about trans fatty acids intake and attempt to decrease their dietary intake because of the high risk associated with consuming these. They can lead to cardiovascular diseases because of HDL, because of this high LDL or cholesterol levels being high. So now, what is important is for us to look at the nomenclature. The nomenclature, these fatty acids have trivial names. First of all, the systematic names are derived from parental hydrocarbons. They can be saturated or unsaturated, whereby if you're naming the saturated ones, you can have the ending. Okay, here, for instance, this is palmitate or hexadecanoate. So this ending anoate can be inoate if it is unsaturated because of the double bond. Now, the position of the double bond also matters when you're naming. You can either count from the omega side or count from the carboxyl group in the delta system, as we shall see. So we can have a geometric configuration of cis or trans being indicated in the naming. So for instance, a palmitoate can be called a cis delta 9 hexane disonoate. This implies that this is hexadecanoate, maybe there are 16 carbon atoms, and the triple bond, the double bond is at position 9 and it is a cis. So we indicate both the geometry, the position of the double bonds in the delta system especially. We actually have two naming systems, the delta and the omega. The delta we count from the carboxyl group. From here, the carbon that carries the functional group takes position 1, and then you come here up to 9. So this will be the benzene, in the delta system. This will be cis, because all the carbon and all the hydrogen bonds will be on the same side. And then this is delta 9 because it is at position 9 from the carboxyl group. Then in the omega system, we count from the methyl group. So you count from methyl group takes position 1 up to when you see the first double bond, and that will be omega 7. Accordingly, from here there are 7 carbon atoms. So these are different compounds and their namings. Okay. Now we also have the essential fatty acids. Now, yes, we can look at all these saturated and unsaturated, but it is also important to know the essential fatty acids. These are acids that cannot be produced by the human body, and so they have to be synthesized, or they have to be taken in by people, or they have to be taken in the body through diet. Insufficiency of these essential fatty acids can lead to scalp dermatitis, growth retardation, and cystic fibrosis. So essential fatty acids are precursors of docosahexaenoic acid and eicosapentaenoic acid, which manufacture these thromboxanes, prostaglandins, and leukotrienes. Now, what you need to know is that these are very important in a way that they regulate immune system, they regulate blood clotting, they regulate inflammation, and they also regulate blood pressure. So these are other important polyunsaturated fatty acids. We have the arachidonic acid, we have the eicosapentaenoic acid, we have the docosahexaenoic acid. So these ones present in animal tissues are precursors of eicosanoids. These are present in fish, all precursors of different class of eicosanoids present in fish. These ones are present in fish. So these are food sources of omega fatty acids. Okay. Now the next time we shall talk about conjugated linoleic acid in detail. But what we need to know is that these omega-3 fatty acids can be found in fish. They are high, most of it is found in fishes, omega-3 fatty acid. The composition is high as compared to other foods. So, thank you. So next time we come, we shall talk about conjugated linoleic